So I am Mohammed Ibrahim. I'm a postdoctoral fellow in the immunology department at Moffitt Cancer Center, and now we are here at the AACR annual meeting 2023 in Orlando, in Florida. So here's my poster, which is we are working on our lab with studying the effect of the Sotorasib, the selective G12C K-RAS inhibitor, and how it can modulate the tumor microenvironment through increasing the TNFα and interferon signaling and the immunotherapy response. So here by the flow cytometry, we see an increase in the T cells in the subcutaneous tumors and also increase in T cells in the lung orthotopic mouse model. And by the single cell RNA-seq, we found that Sotorasib increased what we call an immune-active tumor population, which is enriched by the TNF and interferon signaling. In vitro, we found also again this results by the treating the cells in vitro. We found an increase in the immune-active tumor clusters, and these tumor clusters are being likely targeted or killed by the TNF and interferon cytokines, as shown here by the single cell and also by the PCR, with an increase in the CXL10 chemokines and also in the MHC molecules. Interestingly, we found that these immune-active tumor clusters that are significantly expanded in the Sotorasib-treated tumor are significantly reduced or shrinked in the Sotorasib-resistant tumor and also in the NSG tumor-bearing mice or a T-cell excluded tumor microenvironment, indicating that this Sotorasib immune modulation dependent on the T-cell interaction with the tumor cells in the tumor microenvironment and also in the tumor cell adaptation. And lastly, we are now working on the Sotorasib effect together with the CAR-T therapy. So we found that when we could treat the cells, the tumor, the human non-small cell lung cancer cell with the Sotorasib together with the CAR-T, we see a significant reduction in the tumor cell viability, and also this dependent on the TNF and interferon, as shown by the co-treatment with the TNF and interferon neutralizing antibody. More interestingly, that Sotorasib, as compared to the Trametinib, which is a global MEK inhibitor, doesn't have an inhibitory effect on the CAR-T cells proliferation, as shown here by the viability and also by the CD69 proliferation marker. So the take-home message that Sotorasib transforms the tumor cells into a more dominant, less proliferative, but more immune-active phenotype through the MHC expression, high expression of the MHC and high expression of the chemokine molecules to attract the T cells, secrete the cytokines, and kill the tumor cells. Thank you.